Thank you for the introduction, and thank you for the conference organizer to provide the opportunity for me to present our new method TREE-QMC to estimate species tree from gene trees use quartet graph construction and the graph cut. Um, Species tree estimation is useful and interesting because the species tree tells us how life evolved on the Earth and how genetic diversity is formed during the history and how the species adapt to their environments. In our problem setting, the input is a set of biological sequence labeled by a set of species, and the output is a non-rooted tree whose leaves are also labeled by the same set of the species to represent the evolution history of the species. Although we have an abundant amount of biological data, which makes it possible to accurately estimate the species tree, we found that species tree estimation could still be challenging because of the gene tree heterogeneity, as we follow the following hierarchical model of genome revolution, where we have given any species tree, we first can generate a set of gene trees evolving inside the species tree following certain models, for example, multi-species coalescent model. After that, we can then generate a set of alignments for each gene tree, also, for example, using the generalized time reversible model. Then we get a set of biological sequence, which is in reality the observation or the input of our method. Note that the gene trees could be different from each other and different from the original species tree due to the certain biological processes like incomplete lineage sorting. And on the other direction, given when we observe a set of biological sequence, we may first estimate the gene trees and then summarize the gene trees using a certain method to estimate the species tree. Many popular summary methods are based on a quartet, where a quartet is an unrooted tree over a subset of 4 leaves or 4 species, including the method Astral and the other method based on a set of quartets like wQMC. Note that as a summary method for gene trees, if we wish to work on the quartet set, the number of quartet set could, the size of the quartet set could be up to n to the 4th power, which is huge. We use weighted quartet consensus as a solution to the species tree estimation, where we try to maximize the number of the quartets displayed in the estimate species tree that is also shared by the input data set. Weighted quartet consensus is good because the optimal solution to this problem, if there are no errors in the input, is a consistent estimator under the MS model. That is, the error goes to 0 with high probability as we feed more data into the methods. However, this problem is NP hard, so a set of many heuristics has been developed to solve the problem. Our method TREE-QMC seeks to address the following 2 challenges. First, we want our method to be fast, that is, it is scalable to a large data set with large numbers of genes or a large number of species. I also want it to be scalable to very different input gene trees. Second, we want it to be accurate, especially when there are errors in the input gene trees. Our method is built on the quartet maxcar framework, which is a divide and conquer framework proposed years ago. It subdivides the input taxa set into smaller sets by seeking by partition of the set using graph cuts. Note that a concept of artificial taxa is introduced. There is a divide and conquer process to represent the taxa in the other side of the divide and conquer, so that we can merge the trees after solve the subproblems according to the place of the artificial taxa to get the final solution to the current problem. So artificial taxa is essential to the QMC framework. The key step of the QMC framework is to seek by partition to subdivide the problems by using a quartet graph and max cut in the graph. The definition of the quartet graph is as follows. Each quartet in the input set contributes 6 edges to the graph. 2 of them are called bad edges, where we do not want to cut. 4 of them are good edges, which we want to cut. Taking this figure as an example, we find that the number of the bad edges between A and C is 1 because there's only one quartet that A and C are siblings in the quartet. On the other hand, the number of the good edges between A and B is 2 because there are exactly 2 quartets that A and B are not siblings. After getting the information of the bad and good edges, we want to combine them into a graph by a linear combination of these 2 matrices, where the weight of the edges between any pair of the taxa is given by the number of the good edges minus alpha times the number of the bad edges, where alpha is a hyperparameter can be optimized by binary search. So in this example, we get the cut of A, B, C versus D, E, and the algorithm continues with the 2 subproblems on these 2 subsets. When using QMC as a summary method, there's a problem that we have to work on the input set as a gene trees. So we have to extract a set of quartets from it to construct graphs and perform graph cuts. As mentioned before, the number of the quartets on a subset of n taxa could be up to n to the 4th power. So QMC is not quite efficient as a summary method. We address this problem by directly construct the graph from the gene trees, not the set of the quartets. Take this figure as an example. Suppose we wish to count the number of the bad edges between 0 and 17 in this gene tree. Instead of pairwisely counting the pairs, the other pairs of the taxa to form the quartet along with 0 and 17, which takes quadratic time, we remove the path from between 0 and 17 from the gene tree, and we found that we have 6 subtrees after the removal of the path. We observe that as long as we can choose 2 other taxa to form the quartet from the same subtree, they will be siblings, and a bad edge must be formed. So we can look at the size of these 6 subtrees and choose 2 from each of them and add the binomial coefficient together, which makes algorithm more efficient. The algorithm could be more complicated than I just introduced when there are artificial taxa, because artificial taxa represents a set of input taxa. However, this algorithm can be done in order of s squared times n times k time, which is efficient, where s is the size of the subproblem, and n and k are from the size of the input dataset. Finally, TREE-QMC has a complexity of the n squared times k, which is more efficient than n cubed times k, which is more efficient than n to the 4th power times k if using original wQMC method, when assuming the problems are subdivided nicely. Now we introduce the evaluation of our method in terms of the efficiency. In our simulation study, we follow the generative model to generate sequence data from model species tree. Given any model species tree, we can generate a set of gene trees involving inside species tree, and for each gene tree, we can simulate a set of alignments. Then, given the alignments as the input for the method, we estimate gene trees for each alignment. Then we run methods, get the final estimate species tree, and then we can compare the estimate tree with the ground truth to get the error. In terms of the number of the taxa and how this number affect the efficiency and effectiveness of the algorithm, we found that our method TREE-QMC is not only more efficient than the method based on a set of quartets, it is also more efficient than the actual method. We observed that for the accuracy, it is also more accurate, especially there are more taxa in the data set. As you see from the previous figure, TREE-QMC has 3 versions, and 0 and 1 and 2, and this is corresponding to the normalization scheme of TREE-QMC, which helps improve the accuracy of the method. We found that in original QMC framework, artificial taxa are upweighted because they represent a set of input taxa. Take this divide and conquer process as an example. When we subdivide the problem until we reach a current problem of 6 taxa, 2 of which are artificial, like Y and Z. In this input gene tree, Y represents 3 other input taxa, and Z represents 4. If we just do the naive counting, we found that the quartets involving artificial taxa Z and Y, in total there are 12 of them, while the number of the quartets on the taxa, number of the quartets on the input taxa is always unique. So artificial taxa are upweighted in QMC. One way to easily deal with it is that we enforce the total weight for any 4 tuple to be 1. So we, instead of assign 6 and 6 to the graph, we assign 1/2 and 1/2 according to the frequency of the quartet. This is the first normalization scheme we introduced. We also introduced another non-uniform normalization scheme according to the divide and conquer process. We found that Z represents 4 taxa, 0, 9, 6, and 7, but 0 and 9 are first relabeled by the other artificial taxa X, so they are more distant from the current subproblem, so we assign a lower weight to 0 and 9, but X should have the same weight as 6 and 7. So the total weight for the topology formed by the quartet from the 2 Z's on the right side has lower weight because it's actually relabeled twice. Now we introduce our evaluation of the TREE-QMC method when we apply normalization. We found that our TREE-QMC method is more accurate, especially when there are errors in the estimated gene trees. But when there are not many errors in the input gene trees, the all method just perform as well. We also study the performance of the algorithm when the gene trees are different from each other. We found that the execution time of TREE-QMC is stable no matter how gene trees are different or similar to each other. But Astral has an unstable running time, and it takes more time when the trees are different from each other because the search space of Astral grows a lot when the gene trees are different from each other. We also found our algorithm is a little bit more accurate when the gene trees change. Finally, we study the performance of the algorithms on real biological data set. Here we use a data set of avian species of 48 taxa and up to 14,000 genes. We found that TREE-QMC is more efficient than Astral in terms of the running time, where TREE-QMC only takes 1 minute to finish the data set of 3,000 genes, but algorithm Astral will take about an hour. We found that TREE-QMC recovers a tree that is closer to the reference tree by concatenation, while Astral only recovers 7 clades out of 10. Our method recovers 8 clades out of 10, while the reference tree by concatenation recovers 9 clades out of 10. Finally, we want to conclude that our method TREE-QMC is scalable to the large data set, or gene trees are different from each other, and it is more robust to the errors from gene tree estimation. The future work includes that we want our method to be able to deal with practical scenarios like there are missing data in the gene trees, or there are polytomies in the gene trees that the gene trees are not fully resolved. We want to thank our lab members and friends for the help during the research project, and we also want to thank the reviewers for the valuable suggestions to help us revise the paper. Finally, we want to thank the funding and support from the state of Maryland as well as CBCB at UMD. Thank you for listening.
Okay.